Welcome to AP Biology. This video is a review of protein synthesis. The DNA that exists within the nucleus of each cell is the blueprint for the creation of your body's proteins. According to Beadle and Tatum, each gene is the blueprint for the creation of one protein within the cell. We have a problem though. The DNA exists within the chromosomes that are present inside of the nucleus. The proteins are going to be made at the ribosomes, which exist outside of the nucleus. So somehow we have to get the information that's present within the DNA over to the ribosomes, which are found outside of the nucleus. The bridge between the genes and the proteins that they code for is RNA. The first step of protein synthesis is called transcription. Transcription is going to take place within the nucleus. During transcription, an enzyme called RNA polymerase is going to read a gene, and using the code in the gene, it's going to transcribe a sequence of messenger RNA. When that messenger RNA is transcribed, it will first be processed, and then it will leave the nucleus to go to the ribosome. At the ribosome, translation is going to take place. The ribosome reads the messenger RNA and translates it into a sequence of amino acids. Remember, the sequence of amino acids makes up a protein and determines the shape and the structure of a protein. If we look at the DNA, here we see the template strand of DNA. Remember, DNA is double-stranded. Only the template strand gets read. The other strand is called the coding strand, and that's not the strand that's read. DNA is read in triplets, sequences of 3. The triplet that's being read is called the reading frame. During transcription, RNA polymerase is going to read the triplets of DNA in the template strand and transcribe it into a sequence of messenger RNA. The messenger RNA is complementary to the DNA, except that it has uracil in the place of thymine. So A gets transcribed to U, C gets transcribed to G, so on and so forth. If you look at the template strand of DNA, you can see here it's being read from 3' to 5'. So during transcription, which is happening in the nucleus, the template strand of DNA, the gene, is transcribed into a sequence of messenger RNA. Messenger RNA is also read in triplets. We call them codons. So each triplet of DNA is transcribed into a codon of messenger RNA. The messenger RNA then leaves the nucleus, goes to the ribosome, where translation takes place. During translation, the ribosome reads the messenger RNA, and each codon is translated into a specific amino acid. A sequence of amino acids is going to create the primary structure of a protein. Here we see the dictionary of the genetic code. You can see what codons go with what amino acids. You can see here that there's lots of redundancy in the genetic code. Some important codons. Number 1 is AUG. AUG is the start codon, and that corresponds to the amino acid methionine. Here we have several stop codons, UAA, UAG, UGA. Stop codons are important, so is the start codon. This is how the ribosome knows where to begin translation, and this is where the ribosome knows how to end translation. Now we zoom into those processes. Right here we look at a eukaryotic gene. Here's a template strand of DNA, and right before the functional gene, we have a sequence of non-coding DNA that we call the promoter. The promoter is a control element. It's not transcribed, but it controls whether or not transcription could take place. The first 4 bases of the promoter are called the TATA box because of the bases TA, TA. The TATA box is important because transcription is going to begin when a transcription factor binds to the TATA box. We'll look at transcription factors in more depth in a second, but many hormones within your body, like steroid hormones like testosterone and also other chemical messengers like growth factors, are going to work as transcription factors. They bind to the TATA box. Once a transcription factor binds to the TATA box, then RNA polymerase is able to go and bind to the promoter. So transcription is first initiated when a transcription factor binds, then RNA polymerase binds to the promoter. Once RNA polymerase binds to the promoter, it can then transcribe the template strand of DNA into our sequence of messenger RNA. When transcription is complete, before the messenger RNA can leave the nucleus, it needs to be processed. Several things need to happen to it. Number 1, we add on a 5' cap, which is made up of GTP. We add on a poly A tail, which is a long string of adenine, and RNA splicing takes place. Spliceosomes are going to come in. They're going to cut out the introns. Introns are non-coding, and splice together the exons. The exons are coding. Once the messenger RNA has been processed, it can leave the nucleus. It could go to the ribosome. At the ribosome, translation is going to take place. Translation is going to utilize tRNAs, transfer RNAs, and tRNAs bring the amino acids over to the ribosome. Each tRNA is carrying a very specific amino acid. Each tRNA also has an anticodon. Here you see the anticodon on the bottom of the tRNA, and the anticodon is complementary to the codon. Each tRNA has a specific anticodon that responds or corresponds to a very specific amino acid. The ribosome is going to read the messenger RNA like a conveyor belt. And as the different codons enter the ribosome, their corresponding tRNAs are going to enter with their amino acids. As each tRNA enters with its amino acid, a peptide bond is going to be created between the amino acids through dehydration synthesis. So as the ribosome reads each codon and each tRNA comes in with its amino acid like a conveyor belt, this long string of amino acids is created. This continues to happen until the stop codon enters. When the stop codon enters, a release factor is going to enter the ribosome, which is going to cause the release of our polypeptide. This polypeptide makes up the primary structure of a protein. A large part of protein synthesis revolves around controlling the process, controlling gene expression. Remember, if a gene is being expressed, that means it's being used to build a protein. A large portion of our DNA is non-coding. It's not used to actually code for our proteins, but is used instead to regulate the process of protein synthesis. We call all of these areas control elements. The promoter is one. You have control elements close to the promoter called proximal control elements. And you have control elements really far from the promoter that are called enhancers. These control elements work by having transcription factors bind to them. There are 2 types of transcription factors. You have activators and you have repressors. Activators turn genes on and cause genes to be expressed. where inhibitors, or repressors I should say, are going to inhibit genes, turn them off. Like I said before, many hormones in your body, like steroid hormones, are going to act as activators which initiate the process of transcription. After a gene has been transcribed and it's been made into a piece of messenger RNA, a couple things could happen to the messenger RNA. Number one, the processing. If we want to stop the process of protein synthesis for whatever reason, the cell will not process the messenger RNA. It's one way to kind of put the messenger RNA into a holding pattern. Another way that we can affect the messenger RNA is after it leaves the nucleus. We have another whole group of RNAs called non-coding RNAs. There are two types, one called the microRNA, miRNAs, and a second one called small interfering RNAs, siRNAs. And these two types of RNA have the same job. When the messenger RNA leaves the nucleus, these guys can bind to the messenger RNA, and they could either block it, kind of put it into another holding pattern, or they could actually break it down. It's another way to control and regulate the process of protein synthesis. Here we see all the different levels at which gene expression can be controlled. You can control gene expression by number one, turning genes on or turning genes off, using either activators or repressors, both transcription factors. You could stop protein synthesis by not processing the messenger RNA. You can stop protein synthesis by blocking or degrading the messenger RNA. And finally, you could actually break down the protein after it's been made. Proteins that are going to be degraded or broken down get sent to an organelle called the proteasome, and the proteasome breaks down the protein. In addition to regulating protein synthesis, we can also control gene expression through the structure of the chromatin itself. We can do this by affecting chromatin structure. If we want to activate genes or turning genes on, we acetylate the histones. Remember that the histones are these proteins that the DNA gets wrapped around. By acetylating histones, it opens up the chromatin and it gives RNA polymerase access to those genes. So it's going to turn it on. Chromatin that's loose and open and activated is called euchromatin. If we want to silence genes, if we want to turn them off, for example, create a bar body, you would methylate the DNA, add a methyl group, and this causes the chromatin to become much more condensed. It's going to silence those genes. This really condensed chromatin is called heterochromatin. Any type of inheritance that does not depend on your genetic code, but is instead determined by what genes are activated or inhibited, or in other words, whether the chromatin is open or closed in that region, is called epigenetic inheritance. If we look at the difference between identical twins, the difference between identical twins is because of epigenetic inheritance. They have the exact same genetic code, but the differences that you see in their traits is due to what genes are activated and what genes are inhibited through these two factors. Finally, the last thing for us to talk about are operons. Operons are found only in bacteria, and bacteria use operons to regulate their genes. An operon is made up of the genes of interest, the promoter that RNA polymerase binds to, and another region called the operator. The operator is going to control whether or not RNA polymerase actually has access to the genes. So the operator can give RNA polymerase access, or it could block RNA polymerase. Way upstream from the operon is a regulatory gene that makes a regulatory protein. Right, we call it a repressor. So our first operon is called the tryptophan operon. It's a repressible operon. Bacteria normally produce the protein tryptophan. So this operon is normally turned on. The repressor is normally not bound to the operator, and RNA polymerase has access to these genes, and the cell just keeps making tryptophan. If the bacteria has enough tryptophan, it doesn't need to waste energy to keep making it, so it wants to turn off the operon. Tryptophan is going to bind to the repressor and change its shape. And when the repressor changes shape, it allows it to bind to the operator and physically block RNA polymerase from being able to get to those genes. So the tryptophan operon is normally turned on, and the bacteria has the ability to turn it off. When the tryptophan runs out, it gets released, and this operon will turn back on. The last operon is called the lac operon. This lac stands for lactose, and it produces the enzymes that the bacteria needs to break down lactose. This is an inducible operon. It's normally turned off. So normally speaking, the repressor is bound to the operator, and RNA polymerase does not have access to the genes. However, if the bacteria consumes lactose, it then needs to be able to produce the enzymes, you see right here, that are needed to break down the lactose. So lactose is going to work as an inducer. It binds to the repressor and changes its shape to release the repressor from the operator, giving RNA polymerase access to those genes and allowing those genes to be transcribed and to make the enzymes needed to break it down. That concludes this video.